So this is the next topic in our first module for test 1, and this is microscopy and cell structure. And remember, when I say prokaryote, which two domains are we talking about? Which two domains? Bacteria and archaea, right? And for eukaryotes, remember, just one domain, eukaryote, right? That one's nice and easy. Okay, but lots of different types of organisms fall under eukarya, just even like ourselves, and lots of microscopic organisms. Most of what we know about prokaryotes is about bacteria, though, right? But we will sometimes make a specific designation between bacteria and archaea. But don't forget, when I say prokaryotic cells, I'm talking about both bacteria and archaea, okay? Anybody remember the defining characteristic of bacteria, something they have that other cells don't? A particular type of cell wall, yep. Contains what substance? Yeah, it's a tongue twister, huh? Peptidoglycan, right? So that's one of the things I really want you guys to focus on when you, for homework, watch the gram stain animation, right? By Wednesday class, I want you guys to watch it. It's just a few minute quick animation. So I'm going to jump to slide 22. What did I do? Okay, I hit the wrong button. So we're going to start with this, these two objectives, and that is to identify the principle of the acid-fast stain. Now, one of the main reasons why we stain bacteria in the microbiology lab is so that we can see them. Most of them are relatively transparent. They're going to blend in with the environment. So to be able to visualize them, we stain them. Some stains, like the gram stain, do more than just that. They don't just stain them so that we can see them. They also tell us a little bit more about the organism. So in the case of the acid-fast stain, it tells us if the organisms have a particular type of cell wall that contains a substance known as mycolic acid. There are two diseases that you may be aware of that are caused by acid-fast bacteria. These bacteria fall into a particular genus called mycobacteria. So if you have a bacteria, you know you've got a bacteria, and you stain it with the acid-fast stain and it comes out acid-fast, then you know you're probably looking at a mycobacteria. So in that case, too, one of the major reasons we look at it is the type of diseases that these organisms can cause. Now, don't panic. If you're in microbiology lab, the mycobacterium we use is not one of the major disease causers, right? It's non-pathogenic. So anybody know Mycobacterium? Ever hear that genus name Mycobacteria before? And know what disease it might be linked to? As little as 10 bacteria could give you this infection. Tuberculosis. Mycobacterium tuberculosis. And one you probably haven't heard about in a long time, but there's still cases even in Louisiana every year, is Mycobacteria leprae, right? Which the PC name for leprosy now is Hansen's disease. It's still leprosy. Tuberculosis, I have it on here. So we stain organisms, and these guys are really hard to stain because of this waxy substance, which I'm realizing I don't have the name of it. Shame on me. Mycolic acid. I'll add that in. So the high lipid concentration in the cell wall prevents the uptake of the dyes, and the actual substance is mycolic acid. And that's where you can imagine part of the reason why they have the name they do. So this is a differential staining procedure, like the gram stain that you guys are going to be learning about. It has multi-steps. So they do the first stain, which is carbofusion, And you'll see this pinkish color, this fuchsia reddish purple color. Those are the acid-fast organisms in this picture, and they tend to be rod-shaped, what we call bacillus. They also clump together because of this lipid substance that makes up their cell wall. So they'll stain, and they stain so well that the decolorizer that we use in this procedure is an acid alcohol, and that's partly where this stain gets its name, acid-fast. So even the fact that we use hydrochloric acid and alcohol, you cannot decolorize these cells once we stain them. They stay colored with the carbofusion, this purplish pink fuchsia color. So acid, referring to the fact that we're using acid fast as in it stays, not as in fast, going fast. So then they use another dye to stain the non-acid fast or the acid fast negative cells. Those guys are decolorized by the acid alcohol. So they would be colorless, you wouldn't see them. But we want to know if something doesn't stain acid fast, so we need to stain it again so we can visualize it. So we'll use an additional stain, what's called a counter stain. And methylene blue, some other contrasting color. Other procedures call for a brilliant green color. They don't actually look very bright usually. This is actually a really good stain. But you can see there are other cells present on this slide, right? A bluish green color. These would be referred to as non-acid fast or acid fast negative. So those aren't the ones we're concerned about, right? It's the ones that stain acid fast. And the two diseases, or the genus, is Mycobacterium. Oh, I thought I had the two diseases on here too. Shoo, shame on me. So Mycobacteria tuberculosis, right? So the disease would be tuberculosis, and leprosy, Mycobacteria leprae. Let's see if I put it in my notes. Nope. Shame on me. I know I'm spelling that wrong. There we go. Tuberculosis. Let's see. Or you'll see it as Hansen's disease. There we go. So bacteria, and notice I said bacteria, as opposed to just prokaryotes in general, although most prokaryotic cells will have these shapes as well. Their cell wall and their cytoskeleton help them hold these specific shapes. So in addition, when they divide, sometimes they'll stay next to each other in what we refer to as arrangements. So there's lots of terms, unfortunately, for you guys to memorize, related to the shape of the organism, and then if they stay next to each other, the different arrangements after they divide. So the rigid cellular component that helps prokaryotes hold specific shapes is what? The cell wall, right? The cell wall and their cytoskeleton. In this case, notice I said prokaryotes. So that's both bacteria and archaea. Where we could get more specific, right? We could talk about the exact chemical composition of bacteria. In that case, it would be its peptidoglycan. So in addition to the gram stain, I also have an animation that shows the different shapes of bacteria that I'll post for you guys to be able to watch. So a coccus is a single spherical shaped bacteria. Right? Three-dimensionally, it would look like a sphere. If we're talking about more than one, we say cocci. So in this picture, you see several cocci. So this is Latin. I is plural, us is singular. I just always remember it's the opposite of what we're used to, right? When we usually say I, we're talking about a single entity ourselves. When we say us, we're talking about a group. In this case, it's opposite. Us is the singular term, coccus is singular, cocci is plural. So this next shape, right, to relate it to something that we're aware of is like a sausage or a hot dog shape, right? Or if you're a candy person, Mike and Ike's, right? Or my favorite, hot tamales. Okay, then you can imagine that three-dimensional shape, right? It's sometimes just referred to as a rod, but the technical term would be bacillus. Oops. I guess I don't have the right animation thing up. Okay. The problem with bacillus is it's also a genus name. So what would be a dead giveaway other than reading the content of a sentence that would let you know that we're talking about a genus name as opposed to just the shape? It would have two names. It would have the genus name and the species name. And what about how that would be written? It would be italicized if it was typed, right? And it would be what if you handwrote it? It would be underlined, right? And then the contents of the sentence would let you know too. Can't go by the capitalization though, because it's at the beginning of the sentence, you'd still have to capitalize it, right? And then of course, plural for that would be bacilli, right, instead of the US. This one is really short rods. And so sometimes in three-dimensional space, they may look, depending on what angle you're looking at them, they almost might look round. So they've kind of combined the two shapes of being spherical and being rod-shaped in this name and saying coccobacillus. And then, of course, if it's multiple ones, it's coccobacilli. This next one, again, notice it's an overall rod shape, but it's not straight. It has a little bit of a curve to it, almost like a comma. This is vibrio, or vibriose. This is what we're used to, just add an S, right, if we're talking about more than one. This one's also a genus, right? Vibrio cholera, right? The causative agent of cholera is a vibrio. You'll also notice in this picture over here, right, you see this little line coming off, these two? That's a flagella, right? It's motile by means of flagella. And it's very common for this shape of organism to have flagella. The next one is spirillum, or spirilia, an A instead of the UM for multiple ones. And this is a long rod with multiple curves, as you can see. This picture, too, you can see that it has several flagella at both ends. Right? All these little hair-like things, those are flagella. The next one, three-dimensionally in three-dimensional space, it would look like a slinky or a spiral staircase. It's helical. So it's a long rod, right? But it corkscrews. And this is your spirochete or spirochete. And again, just spirochetes, add an S, or you're talking about more than one. This unique shape and its unique ability to move is directly linked to its axial filaments. You see these black lines running through this electron micrograph? Those are like flagella, but they run along the outside of the cell. encased within an outer sheath. And they've actually genetically engineered spirochetes to not produce the axial filaments, and then they just end up being this long rod. They cannot hold this shape of being helical without those axial filaments. They also cannot do their characteristic movement, and that is that they're able to corkscrew through their environment. Right, so they kind of twirl along. The animation that I'll link up for you guys, they do the best they can to show you this. And then I have some YouTube videos where you can actually kind of see these guys moving. The unfortunate thing about microbiology lab is that, especially like spirochetes, to give you an example of one, the causative agent of Lyme disease is a spirochete. The one that causes syphilis is a spirochete. So, of course, we don't want to be working with any of that kind of stuff in lab. These organisms are really hard to grow, and a lot of them are pathogenic. So we don't get to see and play with any of these organisms in microbiology lab. Pretty much all the ones we work with are rods and cocci. That's it. But these other really cool shapes do exist, right? And we do need to be aware of them. And of course, they can be helpful diagnostic tools, right? If scientists look at a sample and they see something like a spirochete, they're like, oh boy, chances are this is bad news, right? These are not part of our normal flora, right? These interesting shapes of bacteria. So what structure do these cells not have? So in this case, these guys cannot hold a shape. So what are they lacking? What helps them hold their shape? Cell wall, right? So these guys are lacking a cell wall. The term that we use for these is pleomorphic. Pleo meaning many, morph referring to shape. So again, there's this exception in the bacterial world. And there's a whole genus of bacteria that lack a cell wall. So therefore don't hold a characteristic shape. So this one is Mycoplasmas, and this is Mycoplasma pneumoniae. This causes a milder form of pneumonia, commonly referred to as walking pneumonia, because people are up walking around and they don't realize quite how sick they are till they overexert themselves, like running up 4 flights of stairs and passing out at the top. One of the resident assistants when I was in college, we raced up the stairs one night when we were doing rounds, and she passed out at the top on me. She had walking pneumonia. Right, you just sometimes don't realize, right, how ill you are. So this one, myco, you notice that we've said myco before, right? Mycobacteria, and partly why mycobacteria, these ones that acid fasting got that name, is because when they grow in culture, they almost look like fungi, right? Because they stretch out, they clump together, they almost look like hyphae sometimes. But as the name implies, bacteria, in fact, they are bacteria. Here, myco, again, think about the fact that they kind of stretch out sometimes, and so they kind of are misconstrued as being hyphae or fungi, right, of being mold. So that's the myco term, because remember, the study of mycology is a study of fungus, right, and molds and yeast. Plasma refers to the fact that these guys just have a plasma membrane, right? They don't have a cell wall, right? So I always think myco, fungi-like, plasma just has a cell membrane, right? A plasma membrane, no cell wall. So that ties into something we're going to talk about later too. How do we kill bacteria? We've learned about penicillin so far, right? What did I say the target of penicillin was? Anybody remember? It has a specific target. It stops the cell from being able to make an importantly structure that attributes to their shape. It's the peptidoglycan. Right? So if these guys don't have a cell wall, do they have peptidoglycan? No. So could I give you penicillin if you have this infection? I could, but it wouldn't do anything for the infection, right? It wouldn't affect these cells because they don't have a cell wall anyways. They're not making peptidoglycan. Penicillin's not going to affect them. So see how knowing about the organisms really helps us in treating our patients, right, and making them better, right? This would be a really bad mistake to give the wrong antibiotic for this type of infection. So remember I said sometimes when they divide, they'll stay next to each other. Some of them are even connected to each other. So in the case of cocci, because in three-dimensional space they can divide in lots of different planes, you can get a bunch of different arrangements for cocci. So when they divide and they stay together in pairs, it's referred to as diplococcus if you have just one pair. And then if you're looking at several pairs, it's diplococci, right? And at one time, I think this was a genus name, but I think they're phasing it out. But to give you an example, again, of where this would be beneficial is Neisseria gonorrhea is a diplococci, right? So if they take a sample, right, they gram stain it, and they see these diplococci, they see these cocci shaped in pairs, right? It's a pretty strong indication that that person may have gonorrhea. If they take a spinal tap and they see gram-negative cocci, it might be Neisseria meningitis, right? So that's a common bacteria that can cause meningitis, an infection of the meninges. So knowing these characteristics about organisms really helps us in the identification of them. And then the next one is, and I have all of them written out on this next slide. See right there. So don't worry about necessarily writing it out. So pictures-wise, if they stay in one plane and they form a long chain, this is streptococci, right? If you have multiple chains or streptococcus. This is also a genus name, right? You've probably heard it before. You've probably heard it abbreviated, strep, right? That's an infection of your throat caused by Streptococcus prurigens, typically. And this is how I remember strepto refers to a chain. I think about how I would not want to swallow a chain when I have strep throat, right? Your throat is so sore, right? And I think about that tube, right, of my throat. And so when I say strepto, I think of chain, right? I think of my throat. I think about that long tube, right, that these would form. Make sense? Okay. Bacillus, rod-shaped bacteria, can also form chains, right? So this would be referred to as streptobacillus or streptobacilli if you're talking about all the chains in this picture. That's about it. Sometimes bacillus will form pairs. It's very rare, but it does happen. Most of the time, the most complex arrangement for rods is just chains, so streptobacilli. For cocci, more choices, right? They could divide in two or more planes, and they could form these characteristic, what we call packets, where it's like a cube structure. Others will form clusters, and this is referred to as staphylococci. Again, probably a genus name you've heard before. Staph infections, an abbreviation of the fact that Staphylococcus aureus probably is infecting maybe your skin or some other area of your body. So if you were to look at staph, right, under the microscope, it would be cocci, right, and it would be in clusters. Staphylo refers to that clustering effect, right, them clumping together. Sarcinia is the name given to that packet that you'll see. And this is also another genus name. Tetrads are also formed, and this is where you have that group of 4. Your chains, as I said, is streptococcus, and your pairs are diplococcus. And again, this really helps us, right, especially when they're trying to identify, right? Particular organisms have particular structures. And as you can imagine, right, strepto were named after the fact that they form these characteristic chains. And the same thing with staph, right? They form these characteristic clusters that whole genus of bacteria. So all cells, right, the definition of a cell is it must have a cell membrane, right? This unfortunately has multiple names, right? Sometimes it's referred to as the cytoplasmic membrane, sometimes it's referred to as the plasma membrane, right, or just the cell membrane. I typically stick to just cell membrane. Makes sense to me. But know that all those names exist. So we're going to look at the functions of the membrane itself. We're going to look at the functions of the protein components, right? The proteins that are embedded within that membrane and their important functionality. We're going to compare bacterial cytoplasmic membranes, again, because that's what we know the most of as far as prokaryotes go, to eukaryotic plasma membranes, right? What are the similarities? What are the differences? And as you can imagine, functionality-wise, they're very similar. They're made up of phospholipids. And this is a very delicate layer. Is a thin fluid structure, which means that it moves. It's not rigid. It's not stuck in place. It surrounds the cytoplasm, hence why it's sometimes referred to as the cytoplasmic membrane. And it really defines that border between the cell and its environment. It actually makes a cell a cell. Without a membrane, you're not a cell. This barrier, as you probably learned in biology classes, is semi-permeable, which means not everything can get in. And some things have to be transported in or even out of that cell. I always consider the membrane as like that picky bouncer outside the nightclub, right? It's only going to let certain people in. Okay, so the same thing with this membrane. Water, gases, small hydrophobic molecules can pass through by simple diffusion. Go from an area of high concentration to low. But other things are going to need help across, right? Facilitated means help, right? To help. So in this case, anybody know what helps things across the membrane? What's the function of those proteins? Those proteins, right, some of them are actually transporters. They even open up pores or help move stuff from one side of the membrane to the other. Right? Your proteins in your membranes are the facilitators. They're helping move stuff across. Active transport, still probably using proteins, but what's the difference here when we're saying active? What do you have to get this to happen? Energy. Right? You have to expend energy in order to move stuff in or out. So there's your proteins, right? And they're embedded in this phospholipid layer. Some of them will completely span the membrane from top to bottom. Some of them can open up, like I said, as a pore and stuff can travel through them. Others act as receptors where they're going to transport a signal from outside to inside the cell. So receptors help that cell respond to its environment. All cells, right, have proteins on their surface that are acting as receptors, allow you to sense your environment. So this is how your cells basically touch things, right? Smell things, see things. It's all molecular based. It's all these proteins and other molecules associated with these proteins in your membranes. And then transport gates, right? They're going to allow stuff to facilitate through from one side to the other. They may even need to expend energy to achieve that goal. These guys are not stuck in a particular location either. They float through this sea of phospholipids that makes up this membrane. So they're almost like icebergs floating on the sea, these proteins. They can move around on the surface of the cell. Mosaic refers to the fact that it's made up of several different pieces, right? We have all these different proteins and different molecules associated with them and the phospholipids. So all these pieces together, though, form this beautiful model, right, that is our membrane. Where when you make a mosaic, right, when you break up tiles and make a picture, it's stuck, right? They glue them to the wall. In this case, it's pieces, right? But it's fluid. It's constantly changing. It's dynamic. It's moving. So for prokaryotes versus eukaryotes, they're similar in structural and function, right? So you got phospholipids, you got proteins. You're defining a barrier. You're determining who's getting in, who's getting out. Those proteins, as we saw, and as I said, are important for transport, and they're important as receptors for cell signaling. Also, some of them can help maintain the integrity of the cell. Can be structural support. One of the main differences in the structural support of a membrane is the sterol-like substances. For animal cells, we contain cholesterol. So your body actually makes cholesterol, purposely puts it in your cells. This is why when you eat other animals, right, if you eat too much, you have too much cholesterol. There are some people that genetically, unfortunately, make too much cholesterol, right, and may have cholesterol issues because of that. So it can be twofold. It can be your diet, what you're eating, what you're putting in your body, and also what your body is manufacturing, because it's necessary for life. One of those differences helps us in combating other eukaryotes, because animal cells have cholesterol, where fungal cells have ergostol in their cell membranes. This is a different cholesterol substance. And because of that, we have drugs that are antifungals that target that ergostol. Either stop the cell being able to make it or to interfere with its structural ability. And again, if a cell can't maintain its membrane, it's going to burst and die. So that's one of the ways that we can destroy fungal cells, is attacking their difference between us and them, right? We can target that ergostol. We don't have that in our membranes, just bacteria, just fungi do. So next time we're going to look at some outer layers, right? But as I said, I'm going to go back to the beginning. So in addition to finishing up your guys registering for Connect and maybe doing that first assignment if you're ready, and finishing up your introduction, I want you to watch a couple of animations I'm going to post for you guys. Yeah, I'm gonna put them on today. Yeah, I'm gonna put them on Canvas. In Module 1, there'll be a link that says animations. Right under here, I'll put it.